Hi guys, so today we are going to start talking about RNA. To start out today, you probably want to split your screen as you've done before so you can see the PowerPoint on one side and the note sheet on the other side. So here we go. All right, so we are starting to talk about RNA, right? So you just had a quiz and we talked a lot about DNA. Right? So we'll get more into this, but DNA is going to be used to make RNA, which is then going to be used to make all the proteins that we need in our body. And that's what we're going to be working on for the next couple of weeks. All right, but let's review what you know already about DNA. All right, what does DNA do? What is its function? Where do you find it in the cell? Where is it located? And what is DNA made of? Hopefully you know these since you've already taken a quiz on them. All right, so what is DNA? What is its job? What is its function? So DNA is like the blueprint for life. It's what makes your traits. It's what makes it has all the instructions for the proteins in your bodies. And a lot of your cell is made of proteins. All right, a big part that's going to be coming up soon, if you remember, a ribosome, which is this little blue dot, little blue organelle inside the cell. All right, we also have them attached here to the endoplasmic reticulum, the rough endoplasmic reticulum. Ribosomes are what make proteins. So the DNA is going to give the directions to make those proteins in the ribosomes. Right? Well, here's our next question. Where is the DNA located? Right? So the DNA is in the nucleus of the cell. Right? One problem though, DNA is too large to be able to fit outside the cell. It's kind of trapped in there like it's in a little jail. Right? It can't come out. Right? And that's where our RNA is going to come into the picture. All right? Because if the directions to make proteins are inside the nucleus, how am I going to get them over here to this ribosome if the DNA can't leave the nucleus? Right? So in order to get our instructions in our DNA out to our ribosomes, we make a copy of the DNA in the form of RNA so that it can leave the nucleus and come out and meet up with our ribosome. Technically, that's called mRNA because that's another difference you'll see with RNA as we're talking today. RNA has 3 forms. DNA just has the one. All right, and then what were the components of DNA? So remember, DNA was made of nucleotides, and we had our 4 bases, right? A goes with T, apples in the tree, so adenine to thymine, and car in the garage. C goes with G, so cytosine goes with guanine. All right, so now we're going to do some new stuff. So talking about the same questions, but this time we are going to talk about RNA instead of DNA. All right, what does RNA do? Where is it located in the cell? What is RNA made of? How is it different than DNA? All right, and then kind of a new question 4 there. DNA, there's only one kind of DNA. RNA, we actually have 3 different kinds. All right, so the function of the RNA. So the RNA is a copy of the information in the DNA, and we will practice with that later. All right, so it's going to be like the bases, like the A, T, C, and G will be copied over into the RNA form. All right, they're going to be used by ribosomes to make proteins. So here's my DNA in my nucleus of the cell. All right, I'm going to copy it and make RNA that can leave the nucleus. It's going to get fed through a ribosome and then make proteins, all right, that your body can use to make new cells or that kind of thing. All right, this is actually called the central dogma, like the main idea of biology. This is how our DNA is used, how our genetic information flows. So our DNA is used to make RNA, right? And then the instructions in that RNA are used to make all the proteins that our body needs to function. Right? So this is the central dogma of biology: DNA to RNA to protein. So make sure on your note sheet that you write DNA, RNA, and protein on those little blanks above the pictures. All right, this is just another example, shows it in a little different way. So we have our DNA, right? Gives us our instructions for RNA, and then our RNA gives us our instructions for proteins. Over the next couple of weeks, we're going to be going through this more. All right, so we have our DNA, right? And if we're going to make RNA here, or sorry, this is my DNA here in red, right? And we're going to meet it up with complementary RNA. So my RNA here is in blue. You will see some differences here that we're going to talk about. RNA does not have thymine. It's going to have uracil, U's instead. All right, and then that's going to be read 3 letters at a time to make code for each amino acid. And we're going to be getting into that in the next few weeks. Okay, so DNA, right, was located in the nucleus, but the RNA can leave the nucleus. So we find it outside the nucleus in the cytoplasm of the cell. It can also be attached to those ribosomes out, but basically we can just say DNA is in the nucleus, RNA is out in the cytoplasm, like the jelly-like substance of the cell. Right? You may have noticed this difference in some of the animations that we looked at before, the pictures. So DNA was double-stranded, right? Double helix. RNA is going to be single-stranded, right? So we're going to have that backbone that we'll talk about in a second, and then we're going to have our bases, right? A, U, C, and G. But notice there's not another side. Like DNA, I have my 2 sides here, the 2 blue. Here I've only got the 1. That is on purpose. RNA is only single-stranded, not double-stranded. Right? What is RNA made of? So DNA was deoxyribonucleic acid. RNA is ribonucleic acid. Right? So one nucleotide of RNA, so DNA and RNA are both made of nucleotides. They're just slightly different. Right? In RNA, it's made with a ribose sugar. So that's where DNA got its name, right? Deoxyribose sugar. RNA, ribose sugar. So they both get their name from their sugar. But RNA has the ribose. DNA had the deoxyribose. Right? They're still going to have phosphate groups. And they're still going to have the nitrogen bases. So sugar, phosphate, base. Just like we had in DNA. However, we have a different sugar. All right, so here's a picture for you. All right, so this is the sugar group for RNA. All right, this is the sugar group for DNA, deoxyribose sugar, ribose sugar. So this is my RNA. This is my DNA. They're the same. Really, the only difference here is this has an oxygen and a hydrogen on this spot. On the carb, they call this a carbon ring. All right, this one has only a hydrogen where that one had. That's the only difference, right? But because of that difference, they have different names and a little bit different properties. Right, here again, this is my DNA, right? Double-stranded. We have the bases meeting up in the center. We have our 4 bases, C, G, A, and T. All right, so I have my bases in the middle, and I have my sugars and phosphates on the outside. So with DNA, that'd be deoxyribose sugar and phosphate, and my bases C, G, A, and T. All right, here's my RNA. Notice, single-stranded. So I still have my sugar and phosphate, but this time it's ribose sugar, phosphate. Ribose sugar, phosphate. All right, and attached to those, I still have my bases. C, G, A, but watch out. RNA does not have T's. RNA only has U's, uracil. All right, so still 4 nitrogen bases, but RNA is going to have U's, no T's. All right, so slightly different base pairing. All right, so we looked at a similar picture when we did DNA. All right, so G will still go with C. C will still go with G. But if we are making RNA, all right, A is going to bond to U, right? A to U to A. Notice there is no thymine, no T's in here. You cannot have a T in RNA. All right, again, another picture. All right, so here I have my sugar, phosphate, right? That makes up the side like this, right? And then I have my bases, G, U, A, and C. All right, so we did this with DNA. We made complementary strands. So now we're going to try it with RNA. So if I start with the RNA, A, G, C, T, A, to make my complementary strand, I have to do the letter that it pairs with. So A in DNA would go with T, but we do not have T's in RNA. So A is going to go with U. G is going to go with C. All right, see if you can figure out the rest. All right, so here we go. If we were making RNA, we cannot have thymine, no T's. A would go with U, G would go with C, C would go with G, T would go with A, and A would go with U. Right, and last but not least, I did mention today with RNA, DNA is just DNA. There's only one kind. RNA, we are going to have 3 types, and all of them are going to play different roles as we get talking about how we actually make these proteins. All right, messenger RNA. When we were talking before about the RNA that leaves the nucleus, that's mRNA, messenger RNA. So make sure you write that on your blank. All right, just like we just did, A goes with U, C goes with G. Complementary strand of DNA is made into mRNA. All right, so that's basically what we just did right here. All right, this would actually be a strand of mRNA. All right, that's what our ribosomes in our cytoplasm, in our cells, are going to use to make proteins. They're going to use the mRNA. It's like the messenger. It carries the instructions from the DNA. All right, we have rRNA, or ribosomal RNA, because it makes up ribosomes. All right, so rRNA actually makes up the ribosome that's going to make the protein. All right, and last but not least, we have tRNA. The T stands for transfer, because it's going to transfer, like carry things. A small piece that transports amino acids. So remember, proteins are made of amino acids. So the tRNA is going to carry or transfer those amino acids to the ribosome to help build proteins. All right, so we will be getting more into this over the next couple of weeks. All right, there's just a picture for you guys. So here's your mRNA, right? Single strand, just like we did before when we went from DNA to RNA. We did A, U, C, U, A. All right, very simple, single-stranded. Ribosomal RNA, it looks kind of funny. It's in this. So it makes a ribosome. So this would be our ribosome. And you can see they tried to show you the RNA inside here. And this is actually going to be a tRNA. So it kind of looks like a T. All right, I would have my bases down here like A, C, G. All right, and I'm going to carry an amino acid up here. Transfer RNA is going to meet up with messenger RNA inside the ribosome, but we'll get talking about that process later. That's called transcription and translation. All right, and last but not least, there are a couple of questions there on your note sheet for you to answer about the difference between RNA and DNA. There's a short video linked in your note sheet. It's like a 2-minute video, and it goes over all the differences with you. And the guy draws them. It's kind of cool. So hope you guys have a good day.